Hello everyone, myself Krupal Patel and today I am presenting my research work that is study on shell utilization pattern of two sympatric hermit crab species on the rocky intertidal region of Veraval, Gujarat, India. Starting from the introduction, hermit crabs are the annumerant crabs well known for inhabiting gastropod shells to protect their delicate abdomen from various biotic and abiotic factors. Hermit crabs require increasingly larger shells throughout their lifespan to maintain shelter and protection from predators, keeping them in constant search of a suitable shell. The selection of a shell is not a random process, and several parameters of the shell affect the selection, including three main factors: shell species, size, and availability. Gujarat is the westernmost state of India, having around 1600 km long coastline, which is 21% of the total coastline of the country. Out of 17 species of hermit crabs reported from the state, Clibanarius, rhabdodactylus, and ransoni are commonly found in the rocky intertidal region of Gujarat state. Till date, no study has focused on the shell use pattern of sympatric hermit crab species in India, and therefore the present study aimed to examine the difference in gastropods' shell use pattern of sympatric species of hermit crabs. So, the present study was conducted in the rocky intertidal region of Veraval, Gujarat state, India. Random sampling was conducted using catch per unit effort method from January to March 2021 during low tide time. Collected specimens were kept in icebox and brought to laboratory. In lab, the hermit crabs were removed from the shell and the shells were identified up to species level. Further, shell morphology like shell total length, shell aperture width, dry width, and volume were measured. In case of hermit crabs, their species were identified and were further categorized into male, non-ovigerous female, or ovigerous female. For crab morphometry, hermit crab weight and shield length were measured and were further sorted into different size classes based on their shield length. Coming to the result and discussion part, total 1000 individuals of each species were collected. The overall size of both the species was similar. However, sexual dimorphism was observed where males were significantly larger than females. Cerithium and ransoni utilized 29 species and 28 species of gastropod shells respectively, which is higher as compared to the gastropod shells used by other hermit crab species. There was extremely high overlap in the utilization of gastropod shells between the hermit crab species. Although the hermit crab species highly overlapped, samples contained mostly either one or the other species. The apparent aggregation of conspecifics could be attributed to temporal niche segregation caused by differences in their diurnal rhythms, conspecific attraction, or competitor avoidance. Ovigerous female of C. rhabdodactylus and C. ransoni occupied 23 species and 14 species of shells respectively, which is very less compared to the shells occupied by males and non-ovigerous females. Several other studies have found similar results, suggesting that ovigerous female are choosy for specific shell having larger internal space suitable for accommodation and incubation of eggs. Among all the occupied gastropods, these 5 species were highly occupied, and among these also Cerithium caeruleum was mostly preferred by both the hermit crab species, as here we can see that more than 60% of the population has preferred Cerithium caeruleum shells, possibly due to the higher abundance of the shell in the study area. Similar pattern of shell preference has also been observed in other Clibanarius species too, suggesting that the abundance of shell play an important role in the shell selection. Here the figure shows variation in different morphological parameters of the 5 highly preferred shell species. It can be observed that Turbobrunius and Lunella coronata are voluminous shells with larger aperture, whereas Cerithium caeruleum and Tangula granulata are comparatively longer. For CC analysis, the individuals of both the hermit crab species were segregated into different sexes and size classes and given a specific code name as shown in the table. CC analysis revealed that the smaller individuals of both the hermit crab species profoundly utilized Cerithium caeruleum and Tangula granulata shells, while larger individuals utilized Lunella coronata and Turbobrunius shells. The more spired and smaller aperture shells of Cerithium caeruleum and Tangula granulata protect the smaller individual from desiccation during low tide in the exposed rocky intertidal region, while the robust and voluminous shells of Lunella coronata and Turbobrunius can protect the larger hermit crabs from other predatory species in lower intertidal region. Coming to the concluding remarks of the study, present study revealed that both the Clibanarius species utilized greater diversity of gastropod shells, probably due to rich diversity of gastropods in the study area. Cerithium caeruleum was the most preferred shell species, as the higher abundance of the shell species ultimately provides more choice for varying size hermit crabs. Ovigerous females used only a few shell species compared to males and non-ovigerous females, as they have specific requirements. There was a high overlap in the use of gastropod shells between the two hermit crab species, with specific preference for 5 shell species, probably due to their higher abundance in the study area. However, there was an evident shell resource partitioning between different sexes as well as life stages, ultimately reducing the intraspecific competition. These are my references. Thank you.